So yeah, thank you very much for coming to today's neuropsychiatry meeting. Just a few sort of housekeeping notes before we start, if that's okay. We are recording it, so be aware if you do want to pop your face on the screen to ask a question at the end, that it is being recorded. We are very excited to have Matthew Noor, who's coming to speak today. He's an alumnus of KCL and SLAM, and has done a bit of a journey around the houses in the last few years. He's been finishing his PhD at UCL with Ray Dolan, and then after that moved to Oxford, where he's now an academic clinical lecturer, working out there, and also finishing his clinical training there. He's maintained links with UCL. He's a postdoctoral fellow at the Max Planck UCL Center for Computational Psychiatry. He's going to talk to us about cognitive and neural map representations in psychosis. While Matt's giving the talk, please mute your mics, switch off your cameras, don't take control of the slides, which always seems implausible, but it can happen. You can ask a question, either writing it in the conversation, and I'll sort of pick those up at the end, or you can raise your hand, but if you could do so at the end of the talk, that's probably easier. So then we can have virtually raised hands and real life raised hands here, and we'll take those in turn. And then if you want to, if I call out your name, you can switch your camera on so we can see you for your question. If you need captions for the hearing impaired, select the 3 dots on the toolbar and then press turn on live captions. Okay, so thank you all for coming. We're going to turn over now to Matt Noor, who's going to be talking about cognitive and neural map representations in psychosis. Thank you, Matt.
Thanks, Tom. Thanks to everyone who's come. Sorry, I'm not going to be looking at the screen, but the audience is in a different place. It's good to be back. So yeah, all of these slides have UCL in the header, and it turns out it's really hard to change the template slide. So I am now at Oxford, but actually much of this work was done at UCL, and that's why that's there. So the title seems might seem quite alien to many who are not coming from this kind of cognitive neuroscience tradition. So I'm going to spend a bit of time sort of sketching out what we even mean by a cognitive map, why it might be a useful way of thinking about many things in psychiatry and in sort of cognitive neuroscience more generally. As a cognitive neuroscientist, I am interested in bridging the gap between brain activity and cognitive phenomena. So how are these implemented in the brain? And obviously that's one thing that neuropsychiatrists care about, perhaps more than any other subdomain in medicine. Often our treatments are physical, whether it's brain stimulation or drugs, and we hope to use those to treat cognitive psychological phenomena. So what is the link between the cognitive level of understanding and the neural level? And my focus is on psychosis spectrum, so I'll sort of flesh that out a bit. And I'm not going to rush that introduction, because I think it's in many ways, these ideas are very interesting and I want to convey them to you. But then I will present an empirical study of something called neural replay in schizophrenia using magnetoencephalography. And so I'll describe why that's relevant to all of the above. And then a slightly more intuitive behavioral test, or a new window into a behavioral test you're all familiar with. What I'm not going to talk about, because there's just not time, are some other things like the relationship to NMDA receptors and DMN dynamics, but this work's been published and there are backup slides if you want to see it. So to start with the kind of main idea, what is a cognitive map and why is it a useful conceptual framework in psychiatry? Really, the notion of a cognitive map, it's a metaphor. So the way in is to think about spatial maps. So when if you think about how we might represent or store information about a city we live in, obviously we're representing information about places, buildings, landmarks, but also about the relationships between them. So how you might get from the London Eye to the Houses of Parliament or Westminster Cathedral or something. So already you can see that within this representation, there's both some notion of what things exist, but also how those things are related to each other. And there's actually reasonable evidence both at the cognitive level and at the neural level, but these things are represented separately. So you might be able to represent the structure of a city more in an abstracted way, or separate from the individual places in the city. And the reason we know about this is because if you spent your whole life in London and then you move to Paris, on day one, you're not going to know any of the landmarks. No two buildings are the same. But actually, very quickly, you might begin to become quite competent at behaving in Paris simply because you know that it's organized in the way that cities are. There's a center, there are suburbs, there are, if you want to get from one suburb to the other, you might have to go through the center. Now, in humans and higher primates, I guess, these same principles apply to the representation of non-spatial phenomena as well. So in the social phenomena, you also have entities or agents, you also have the relationships between them. But now these relationships aren't physical proximity, they might be things like kinship or loyalty. And again, you have similar transferable relational structures. And at the most abstract conceptual level, you can also imagine the representation of concepts. So here, animal concepts, but even emotions or other non-physical, non-spatial phenomena, these will be represented with a relational structure as well. So why might it be, cognitive scientists spend a lot of time thinking about how these things are represented in the brain or in AI agents. And the reason why it's important is because the way that you represent information, where you structure your knowledge, has a huge implication for what you can do with it. So, for example, if a rodent in a T-maze has experience of turning left and turning right, and actually, if the rodent actually represents the T-maze explicitly as a spatial map in its mind, then it can do interesting things like plan routes to desired locations, or perhaps even more interestingly, it can imagine routes that it's never traveled before. It can do shortcuts. And similarly, this competence extends to humans in the social domain. So if you have an organization, if you have a representation of how people are related to each other, again, this is now not physical proximity, but say kinship, then you can plan routes to people through other people. You can make inferences about relationships that you've never directly seen before. And the reason why this is interesting and important for psychiatry or understanding psychiatric phenomena is because obviously we deal with aberrations of behavior, inference, mental simulation. So for example, in worry, you can imagine that, and people have modeled anxiety as an overrepresentation of the probability of a bad outcome in some task domain. Or certainly people who grow up in maladaptive, perhaps environments without good caregiving structures may develop templates for how people relate in childhood that when transplanted to adulthood lead to maladaptive inferences. And actually, although I'm presenting this in a very sort of cognitive neuroscience way, this is basically, in the language of psychodynamic theory, will be an abnormal role relationship schema. So we're talking about mental schemata here and how they might be modulated in psychotherapy. In psychosis, I think the reason this might be interesting, and one thing which always fascinates me about psychosis, is the kind of symptoms that we deal with. So, for example, paranoia, it's really an inference about the way that other people relate to you based on very little information. We have no access to the mind of another person. We often are making inferences about their relationships to us based on very sparse data. So any inferences we're making has to be leveraging some mental schema. So understanding that might be important for treatment. But another phenomenon which is slightly coming at it from a slightly different angle is that phenomenon of thought disorder or conceptual disorganization. This is an interesting cartoon from the 1960s where you probably can't see it, but it's a thought bubble of a man looking at a painting, and you can just see how each thought sequentially triggers the next one. So it starts off by something very glued to the painting, and then that triggers another thought, and an image, and a modification of the image. And what's interesting about this is that if you observe somebody speaking freely, a patient, for example, the sequence of thoughts and concepts gives you some window into their mental organization, potentially. And so this is another window into sort of that, how concepts are structured. So this is all at the cognitive level. At the neural level, how do you begin to tackle this question in the brain? How do you begin to start studying it? And here we can again return to the spatial navigation domain. And for many of you this will be a recap, but basically for at least 50 years, I mean, going back decades before that, we have known that the hippocampus is exquisitely involved in spatial navigation in rodents, as well as episodic memory, of course. And the work of John O'Keefe in the 1970s and before identified these place cells in the hippocampus, which fire when the rodent is in a particular spatial environment, a particular location within a spatial environment. And decades later, the Mosers, Edward and May Moser, discovered grid cells in adjacent entorhinal cortex. So these are cells which fire not when the animal is in a particular location, but they tile the spatial grid, capturing the relationships between spatial locations. And this work won the Nobel Prize in 2014. Adjacent to this work, there was the finding that when rodents are resting, so when they're asleep, in slow wave sleep, when they're eating food, then these place cells sometimes reactivate. They can fire again in the hippocampus. And they don't do so randomly, they do so in spatial sequences. So they place cell for location A fires first, then for location B fires next, and location C fires next. And these locations are close together in space. So you can actually, it's as if the rodent is replaying a real experience of having run through spatial environment, or potentially is even simulating experiences. And the function of this replay is very uncertain, but certainly has something to do with memory consolidation. But more recently, it's been speculated it might actually have something to do with how we build a model of our world, how we build an internal representation of the world offline, during sleep, during mind wandering. And it always struck me that this might be quite relevant for imagining how patients come to very well-articulated, crystallized delusions, for example, of how people relate in their world based on very little actual direct experience. A lot of it may be going on in the offline process, in the process where the brain is kind of in its resting mode rather than its awake mode. And there's evidence now that this has a relationship to the default mode network. Now, these ideas are all in rodents and these are all in spatial navigation, but John O'Keefe himself, with a colleague Lynn Liddell in the 1970s, already wrote a book saying the hippocampus maybe is a cognitive map. It's not just this neural architecture, which we've been studying in the domain of spatial navigation. The same computational principles underlie all relational inference. So not just how you get from point A to point B, but how you get from person A to person B, or the relationship between concepts. This was a very interesting speculative idea, and there was not a huge amount of evidence for it. But in the last probably 10 years or so, there have been increasing efforts within cognitive neuroscience to investigate this using brain imaging. And a lot of the credit here does go to Tim Behrens, who's been pushing this idea, prof of computational neuroscience in Oxford and UCL, but others as well. So I'm not going to talk you through all of the experiments, but just one key one, for example, is, and this is a complicated experiment, so it's just going to get the idea across to you. They asked, this is Alexa Konstantinescu's experiment back in 2016, published in Science, and she asked participants, healthy volunteers in a scanner, to look at birds, and these birds could differ in how tall their necks were or how long their legs were, and by stretching the birds, either the neck length or the leg length, the participants could get rewards. So it's a non-spatial task. However, notice that these birds vary across 2 dimensions, leg length and neck length, so they can be plotted in a 2-dimensional grid, and each bird occupies a location in that grid. And so the stretching of a bird is a trajectory through the grid. Now, the participants are not aware of this, but what Alexa hypothesized was that in order to do this task, maybe there is some movement in conceptual space that the person is doing, and that is underpinned by the hippocampus. And they did the task in fMRI and found some pretty convincing evidence for these grid-like codes, which I won't have time to go into, but they're the same sorts of fMRI signals that you see in spatial navigation, and you really don't see them anywhere else. They're navigation-based signals. My colleague at UCL, Yunze Liu, who's now a professor at Beijing, just a few years ago did another study where in MEG he asked people to learn non-spatial sequences, so how one picture relates to another. Again, this is not a spatial navigation task, and he found that in the rest period after the learning, people, the MEG brain signature showed evidence of replaying fast, rapid sequences at the sub-second scale. as if the same neural processes that underlie replay and spatial navigation might be at play in humans consolidating this non-spatial relational structure. And there's a ton more evidence. You know, I would direct you to Tim Berens' review in Neuron 2018, and his student James Whittington did another one in Nature Neuroscience just this year about what these, you know, trying to consolidate all this evidence in humans. So in psychosis, I've already hinted as to why the phenomenology of psychosis might be indicative of this kind of offline map-like process being implicated. But there's also interesting sort of suggestive evidence on the biological end of the spectrum. Well, before we get to the biology, one behavioral signature of this replay, of this relational inference phenomena, is an ability to infer a relationship that you haven't directly seen. So if you've seen, and this is captured in a task called a transitive inference task or a paired associates task, and rats who have hippocampal lesions can't do this task. Patients with schizophrenia struggle to do this task as well. But a ton of imaging work and electrophysiological work also hones in on the hippocampus in psychosis spectrum disorders, whether that's hippocampal hyperactivity, default mode network dysfunction, GABAergic, glutamatergic dysfunction, atrophy in the hippocampus, but also sleep spindles, which are related to hippocampal phenomena as well. So a lot of this suggestive work pointing that maybe in psychosis there is a problem with hippocampal functioning at rest. And there have been a couple of rodent studies, genetic mouse models of psychosis for what they're worth, but several different ones, 3 convergent models, different genetic perturbations, showing that hippocampal activity at rest is abnormal. In particular, place cell replay is disrupted, but also this oscillatory phenomena which accompanies the replay, this high frequency ripple in the local field potential, is also elevated, is also exaggerated. But there was obviously no work going beyond this. So actually, one of the things that we did, and this is just the first experiment, is to try and tackle this phenomenon in experiments using MEG. And this is what I did during the first half of my PhD. So I'll explain the kind of task outline, the logic, and show you some results. Suffice to say that there'll be a lot that I'm skimming over, but you can ask questions if you want to, or speak to me afterwards. But what we wanted to do was to ask the question if we could get any evidence at all that neural replay, which we think might be located in the hippocampus, during offline rest is abnormal in patients who have had a history of psychosis. And there's really been no effort or attempt to investigate this phenomenon because it's really hard to measure. We until very recently, we've had no way of even thinking about how we could measure this. So I'll explain what we did and why we think it works. Yeah, so this really leverages advances in magnetoencephalography, but not so much in the hardware, but more in the analysis, and I'll explain why. So I'll show you the task in a moment, but the hypothesis based on the rodent literature was that patients would show an impaired replay, potentially an augmented ripple power, oscillatory power, at time points when you think replay might be happening. And this, we wanted to also try and measure directly the neural map-like organization using something called a representational similarity analysis, but I'll explain that later. So the task in its, well, I guess, small sample of patients, half of them are medicated. They're relatively, they're not typical patients you'll see in SLAM. These are people who've had a history of psychosis. Some of them have only had one episode, some of them have had multiple episodes. They're generally well. I mean, they're not at floor when it comes to their PANSS scores, but we're scanning people who have a bit of thought disorder, maybe have some hallucinations every now and again, some paranoia, but generally they're able to consent to the study and they're able to meet me on 2 consecutive days and to do the scan, which takes a bit of time. So the generalizability of these kind of findings always requires some, you know, it's always tricky. This is, yeah, so the task in its, at the highest level involves learning spatial visual sequences, so how pictures relate to one another, which is entirely non-spatial. And I'll explain what the task is in a moment. So participants come in on the first day, and they're acquainted with kind of the rules of a task, which is basically to learn how they're shown pictures of sequences of 4 pictures like this. And they're told in training that the first 2 pictures, the relationship between them is real, and the relationship between the last 2 pictures is real. But the middle pictures, so the airplane going to the headphones, is spurious. Now, the point is that after you've seen a few of these visual sequences, and after they're repeated to you time and time again, you can begin to piece interesting things together. So, for example, in the first sequence, you see that the padlock is related to the airplane, and in the second, that the spaceship is related to the padlock. And actually, if you were to think about this and see it time and time and time again, you would be able to piece together that these actually probably form a chain of 3. So the padlock is related to both. This is not easy to get, but with pre-training, the participants can get it, patients can get it, because you can conceptualize it as you're kind of trying to retell a story, and people understand this quite well. Patients come in on the second day, and we ask them to do the same task, but this time with totally different pictures. So the rules that they learned on day 1 can be transferred, but the identity of the pictures is different. So what this means is that they're having to piece together the pieces in real time on day 2 while we're scanning their brain. And one phenomenon here is there are actually, it's a bit of a small print thing, but when you really piece together all of these pictures into the correct order, there end up being these 2 sequences. So although there are 8 pictures, 4 of them are in one sequence, 4 of them in the other, and these don't interact. So what we can do is we can ask people, after every 5 minutes of seeing these picture sequences, how much they actually know. And what we find is that patients and controls are both very, very good. So for example, here's the kind of question you might get. So you might say, what comes after the banana? And if you look at the bottom sequences, you'll see that the banana is in the left-hand sequence, and it's the 3rd picture. So what comes after it is the house, the 4th picture. Now, this top question is quite hard, because you're being asked to choose between the butterfly and the house. The butterfly comes before the banana, the house comes after. So this is really testing your ability to know within the sequence what is the correct order. This bottom question is a lot easier because the incorrect answer belongs to a different sequence. So all you're being asked to do there is to know which of the 2 families this picture belongs to. And this was actually very, very useful for us because it meant that we could test whether patients show a selective deficit on one versus the other question. And if they showed a deficit on both, they're struggling to follow the task, aren't they? They're struggling to understand the instructions, to pay attention. We found that the patients here in red showed a selective deficit in the sequential learning component, but they were a bit slowed to learn the task in general. But after 15 minutes, after Q3, although both patients and controls had the same matched knowledge, the patients had struggled with this sequential aspect. And we can quantify this numerically. So the reason why this is interesting here is because this within sequence difference, what it highlights is that the aspect of a task the patients are struggling with is piecing together how the observations they are being presented fit together with the correct relational structure into the correct sequence. And that we can now, because we're scanning their brain, we can now ask the question, is there any evidence of of replay of the learned sequences happening immediately after the learning, and whether this is distorted in patients. So the reason we can do this is because after the task, we ask people to sit for 5 minutes in rest, and we're scanning their brain for those 5 minutes. Resting state brain scan and EEG. And there's a couple of other things one other thing in particular to be aware of in this study is that before they do anything, people, both patients and the controls, undergo a very long 20-minute stimulus localizer. So what happens in the stimulus localizer is that every picture they're about to see in a particular order in the sequence is presented randomly. So there are 8 pictures, they're presented probably 40 times each. And the patients and the controls have to do a task just to maintain attention. Now, the reason we do this is because this is crucial for the analysis of how we actually detect the replay. So I'll explain in a little bit more detail. So in this sequence localizer, what we have, if you think about it, is if we're presenting the picture of, let's say, the car 40 times randomly, every time the car is seen, you will get an evoked response in the brain. You will get an evoked response which hits the visual cortex about 100 milliseconds, it sweeps forward, and you have that 40 times, and you have it 40 times for the girl and the hand. So you can train a machine learning classifier to actually predict what the person has seen, given what the brain signature looks like. And because we're using MEG, in fMRI, you would basically have a single snapshot of what the person is seeing at that for the entire 1 second, because fMRI is sampled perhaps once every second or once every 2 seconds. MEG is sampled 1,000 times a second. So we have a snapshot at every 10 milliseconds, in my case, because we downsampled, of what the neural activity looks like. And we can train a machine learning decoder, just a simple logistic regression classifier, to detect every picture versus every other picture based on the MEG signature. And we can find that actually what this is showing here is the ability to actually correctly predict which picture the person is looking at from their brain signature. From 0 being the picture has just come on the screen, all the way to 1,000, which is the picture has been on the screen for a second already. And you can see that at about 200 milliseconds, there's this very sharp peak in decoding ability, which is above chance, which means that if we take that time slice, if 200 milliseconds after you see a picture, we take the snapshot of your brain, I can predict well above chance, nowhere near perfectly, of course, whether you've seen a cat or a house or a hand. And the crucial thing is that we can do this equally for patients and controls. So not everything we measure in patients is abnormal. If it was, there'd be a problem. Probably, but I was worried about this because what if the patients move more? What if the patients are falling asleep, etc., etc. The way we got around this is to select non-university controls. I think it's very, very important. Yeah, I can't stress that enough. You can't get away with not doing that in cognitive neuroscience. You can get away with doing it in PET, I think, in other studies where no one's measuring performance. But in cognitive neuro, the data is going to tell you if your samples aren't properly matched. And so this is perhaps what you might see. So this is averaged over all participants, but the spatial distribution, this is looking at the brain from the top, and each circle is a sensor in MEG. You can think of it like an EEG sensor. These are the weights, the beta weights on the classifiers that are specific to each picture. And you really looking at this, you can't really tell much of a difference, but the subtle differences allow the machine learning classifier to pick this up. So why do we go to the effort of doing this? Well, the reason is because we can now take these spatial patterns and apply them to the data collected during resting state, where the person is not seeing anything on the screen, they're just mind-wandering. And the intuition is that if memories are reactivated in the brain, they're obviously not going to be visually evoked signals, but there's going to be enough similarity, and there's work showing that there's enough similarity that you'll be able to detect the reactivation using these classifiers, very slightly above chance. And so that's what we do. For every time point of the MEG rest data, we apply the family of classifiers. And for every time point of rest data, we can say, here's the probability that the marbles are reactivated. Here's the probability that the butterfly was reactivated. And the difficulty with this is there's no ground truth. You can never know if you're right. You just have to, you're saying, well, this is what happens if I put these two things together. What we can then do, and this is really getting at the heart of the question, we can then say, given this kind of this is a cartoon, this is not real data, given this interesting sort of pattern of activations and reactivations, is there any structure to it? So if the marbles are reactivated, is it more likely for the next picture to be reactivated is going to be something like the butterfly, or is it just random? And if there's no randomness there, and if that randomness is broken across all participants in a systematic way, then that might be evidence that something interesting is happening in the brain reactivations. And to be, if you look at the left-hand side, what I've grayed out there, the important thing to remember is that the rest data that we're looking at this reactivation in comes immediately after a task where people are learning that pictures are occurring in sequences. So all we're interested to see is that, is the reactivation, does it mirror the sequence they've just learned, or is it or not? And if it does, then that's evidence that we've actually induced a sequential reactivation in the brain. And this is, I won't go into the details of how we do this, but essentially this summary plot is a graph which looks like this. This is just a cartoon, but on the y-axis is the evidence that this sequential reactivation exists, and on the x-axis it's the time lag, of the speed of the replay. So over here it's really, really slow replay, and right by the 0 point it's really, really fast replay. So if you see a bump, it means that at this speed there's evidence that replay is occurring. And to cut to the chase, if we do this in the rest data before the task happens, so people come into the scanner, they do a rest session before they've learned anything, then we don't see any replay, which is what you'd expect, because there's nothing to replay, they haven't learned anything yet. But if you do it after they've learned the task, then you see a big bump in the healthy volunteers, which mirrors a finding that my colleague Yunse found in 2019. But this is absent in the patients. And it's always a bit difficult to interpret an absent finding in your patients. So there's a whole bunch of control analyses that we do in the supplementary of the paper to show that it's not just because the signal's in there somewhere, but we just haven't been clever enough about how to find it. We really think there's nothing there. And that this difference is significant. So basically, from before learning to after learning, the control participants show a boosting in this signature and the patients don't. And to kind of recap what this signature is, what in the rodents, this is the signature that we think is underpinning an ability to take experience, to take experience of in the world, and then offline during rest, during sleep, to replay that, to actually build an understanding, furnish a model, an internal representation of what's going on in the world, how things are related. So clearly this is a silly picture sequence task, but the idea is that if this general mechanism is abnormal, that might have implications for how some symptoms are generated, obviously highly speculative. We also see a relationship between the strength of the signature and how quickly participants learn the task. There's some hint that it's behaviorally relevant. Something which we also found is that, and this is an analysis that again I won't delve into too much, but we can ask the question, what is going on in the power spectrum of the neural data during periods of rest when we think these reactivations are happening? And in rodents, what's going on is that you have a very high increase in very high frequency power, so 150 to 200 hertz, what's so-called the ripple band. And for people who, I don't know if people know Yuri Buzaki, wrote Rhythms of the Brain and Brain from Inside Out. His whole career has been on these ripple bands powers. He thinks they're core to cognition. They're also, by the way, well, I guess for this audience, be very relevant because these hippocampal ripples are the most synchronous events in the whole human brain. And they're often what's driving temporal lobe epilepsy. So you have these pathological ripples. So there is this interesting idea that there's some evidence in psychosis, they're also abnormal, just not to the degree that they are in temporal lobe epilepsy. And a large degree of the brain's work in those local circuits is dampening down these ripples through these inhibitory circuit phenomena. Now, when we looked in our sample at what's going on in this high frequency at the time points of ripples, we do indeed find an increase in high frequency power. It's very subtle, and we can source localize it to hippocampus. But interestingly, we find that the patients have even more of this than the controls do. And this aligns with the 3 genetic mouse models, which also found this exaggerated or pathological ripple-like activity in hippocampus in rodents. And also relates to, for example, models of Tony Grace and others about how hippocampal hyperexcitability might drive hippocampal atrophy and that kind of stuff in psychosis. Okay. One final thing I want to show you in this study, which I think is kind of cool, but it takes a bit of, it does take a bit of unpacking, is that, can we do better than this? Can we say, like, all this is going on during rest. This is when the participant is resting. We have 5 minutes of data and we're just looking at what's going on inside. What about when people are looking at the pictures themselves? Can we get any insight about how they are representing these pictures in their neural signature? And here we made sure that in addition to the stimulus localizer at the start, if you look at the top cartoon, remember the stimulus localizer, it's when people are just showing random pictures one after the other. We also included a similar task right at the end, after they've learned the structure. So now what we have at the start of the scan is many, many examples of the brain activity for each picture. And we have the same at the end of the scan, many, many examples of the brain activity for each picture. And the difference, what's happened in the middle, is that people have learned that these pictures are related in interesting ways. So we can ask, does the brain signature change from before to after the task in a way which mirrors how these pictures are actually related in the task itself? So concretely, if you've learned that the marbles come before the butterfly, then maybe in the process of learning, you now actually represent the brain signature for marbles and the brain signature for butterfly becomes subtly more similar. And that's what's called a representational similarity analysis. For me, the more interesting phenomena, which relates to more abstract, higher level cognition, is if you have 2 sequences, sequence 1 and sequence 2, and each sequence has a picture that is first. The concept of first is actually something you have to extract from the data. It's not something that's in the data. It's a cognitive construct, especially the way we present the data. So does the neural signature for the first picture become in sequence 1 become more similar to the first picture in sequence 2? That would be very cool. And the framework we use here is what's called RSA. So I'm happy to talk about it afterwards. But we essentially find that this position code, this abstract position code, does appear to be there about 500 milliseconds into the neural sweep. So what's happening at that time, about 100 milliseconds, you're in visual cortex. By about 200 and 300 milliseconds, you're going down the what and where pathways, the ventral and dorsal streams, and you're in temporal cortex. And around about that time, when you've got these higher level conceptual representations, in the control participants, you indeed see these more abstract relational similarities developing, but in the patients you don't. Now, I think this probably just means that our patients didn't quite grasp fully what it is we were asking them to do to the degree that the controls did, but it's cool that we can find it. And this is a bit post hoc, but you kind of think, well, what if they were just confused though? What if like the patients were doing things in a slightly more intuitive way, which was perhaps not exactly how we wanted them to do it? And we find some evidence that there are neural signatures, for example, what we call these confusion codes, which you can look at in the paper as well. But again, these are things which the patients had more than the controls. So I thought that was quite. It's always quite nice to see that. It means that it's not like you don't have any signal in your neural data at all. Okay, so that's the bulk of the empirical work I wanted to show. And this is all kind of abstract, and it's very dependent on brain scanning. And one of the things I enjoyed doing was doing something a little bit more intuitive. And I'll just show you this for the last, say, 10 minutes or so, and then take questions. So this is to ask the question, at the start you'll recall that I mentioned that one interest was this notion of thought disorder. And actually from my earliest days doing psychiatry, I always found that probably the most fascinating phenomena that you can exist. And one of the things that the intuition behind using speech as a window into sort of cognitive conceptual organization, it goes a long, long way back. So, even William James back in the 1890s said that the stream of consciousness is a bit like a bird's life. So you have these alternating flights and perchings where attention jumps from one concept to the next. And obviously where attention jumps is not random. If you're thinking about your holiday one moment, you're much more likely to be thinking about something leisure related on the next moment, and maybe who you were with, and that sparks off the next thought. And so here we don't have complex brain scanners to tell us what's happening at the neural level, but the behavioral data itself is very rich because it can tell us how one thought triggers the next. And for a long time, this metaphor, the movement of thoughts through conceptual space, might be similar to the movement in physical space, has been around. It's been around in William James, but also it's been around much more recently in neural data. So just two recent papers, could have picked many more, actually arguing, the top one in epilepsy patients with hippocampal electrodes doing verbal fluency tasks, shows that hippocampal theta, which is a spatial navigation signal, tracks semantic distance between concepts. The bottom from Yuri Hasson's group at Princeton, out to just last year, I think. Again, in epilepsy patients with ECOG recording electrodes in Broca's area and Wernicke's area, showed that the kind of neural patterns that people have when listening to podcasts are similar to GPT-2's neural signals. It's wild stuff. It's really cool stuff. And this is what I want to do during my CL, just not with ECOG, unfortunately, but with MEG. But moving away from all the neural stuff, I did something much more simple, which was just simply in the same patients that I showed you to ask them to do a simple category fluency task, something you're all familiar with. Name as many animals as you can, and it won't be won't be surprising to you that patients semantically cluster. They don't just give you random words; they give you words, for example, to start with pets, and then wildlife animals, and then bird animals. Now, if we could only have a way of measuring the semantic distance between words, again, semantic distance, the metaphor to physical distance, then we could actually start analyzing this as a trajectory. And the current word embedding models, ChatGPT, etc., is all the rage now. But I was doing this back in 2019, where these models were already still around. They just weren't conversational agents. So word-to-vec, word embedding models. And what you can do is, many of you, this will be very familiar. You can take any word or any stream of words and represent it as a vector, as a string of numbers. And in this case, the vectors I used are 300 dimensional, so 300 numbers. And these come from models trained on word-word co-occurrence statistics on the internet. So fast text, which is Facebook's model, is trained on 16 billion words. It's a huge amount of data. And the idea there is that which words occur frequently on the internet gives you some indication about how close together those words are semantically. And those 300 dimensional vectors, the more similar those strings of numbers are, the more similar two words are. And so you can quantify this using cosine distance, and you can begin to take a participant's word list and to transform it into number lists, and those number lists into similarity matrices. So here, this participant, cat, dog, mouse, rat, and each color represents how similar these words are in semantic space. Now, the cool thing is that you can take each word that the participant says and visualize it in some interesting, here's a three-dimensional projection. So that list of words that you have is now transformed into a spatial trajectory in space. And once you've got this, then you can begin to analyze it as a genuine trajectory in semantic space. You can start taking these ideas of these metaphorical ideas I showed earlier, you can start taking them more seriously. And because I did these slides last night, I just kind of pasted some results. So I'm not going to go through them all, but the headline, one of the headline results is that this graph just shows that people semantically cluster. So the more the words that people say one after the other are more semantically close than you would expect by chance. That difference is reduced in patients. If you ask them to do another task, which is the letter fluency task, name as many words beginning with P, you don't see this phenomenon at all. And actually, patients and controls are matched, as I'm sure many of you might know, some work being done in the early 2000s, from the 90s to the 2000s, showed pretty convincingly that it was the semantic category fluency task that taxed patients more than the letter fluency task. This is just putting some numbers to it. This is an analysis though, but this is why I took this further to say that let's take the set of words that a participant said. In this case, I believe it is a patient, but it doesn't need to be. And let's draw the spatial trajectory they took. That's what you see at the top, semantic trajectory. And you can see that actually it's a bit all over the place. So there's a lot of zigzagging, there's a lot of doubling back on itself. So you might start by saying cat, dog, then elephant, tiger, then come back and say guinea pig. And you can ask, well, what will be the optimal trajectory through this space? If I was going to visit every word just once, using the minimum distance, what would that look like? And what is the difference in control? And you can see that, I mean, you just have to take my word for it, but there are big differences between the groups, and they relate to performance. And again, you don't see this difference at all for the letter fluency task. So I think one of the things in cogneuro is that it is important to have tasks where your groups don't differ. Otherwise, again, it comes back to the brain imaging data. I won't bore you with going through like all the other analyses here, but essentially they're different windows of onto the same phenomenon. So trajectories through communities, modeling-based approaches, they all show the same thing, that the participants who had a history of psychosis show a reduced ability to semantically cluster than the control participants. And this does relate to the brain signatures that we had earlier in the recon task. So I think I'll end there, just to say that one of the reasons that I've been interested in these ideas for a while now is because I think that they are an interesting way to bridge a brain level understanding of psychiatric conditions or phenomena with an approach that takes seriously the phenomena in their own right, the symptoms, the cognitions, and that at the end of the day, most of our treatments still work by perturbing brain function, whether it's stimulation or drugs. And if we want to develop better treatments, I think we do need a better understanding of how perturbing brain function through the mechanism of cognition leads to symptom generation or not. And I think these questions are also just fascinating in their own right. But yeah, I will let my collaborators, the people grayed out, didn't really contribute to the present work, but did to the overall mission, I guess. Okay, thanks very much. I haven't been keeping track of that, I'm afraid.
This is great. So I think we can still be seen. Thank you so much, Matt. That really, there was a sort of lucidity and clarity to that. I think I genuinely can say I haven't come across in the sort of computational psychiatry talk before, and it's really quite impressive to be on the receiving end of that. So thank you so much. I guess maybe if I can just take the first question, and then if people do want to add questions in the chat or raise their hands, we can take some questions from the audience here. My question is coming back to phenomenology. It feels like, particularly the first work that you were showing us in cognitive maps, that there's a really fundamental concept is that of sequencing and of sort of beforeness and afterness, for want of a better phrase. Thinking about the pathology that I most associate with psychosis is often the kind of causal misattributions of causality. They did, someone did that rather than me, or those sorts of problems. Do you see a way of linking that impaired kind of attribution of causality to something about problems with sequencing? Because presumably attributing causality can only happen if sequences are reliable.
Yeah, I think there's two really deep questions there, and these are things which I was thinking about 3 or 4 years ago, and this guy Dan McNamee, who's a great computational scientist, who's now in Portugal, said to me, stop thinking about causality, because the causality machine learning community is so hard to please that whatever you're going to do, they're going to criticize it. But it's very hard to separate. Causation is not correlation or the rest of it, but actually in the real world, we often infer causation from correlation, and I think even David Hume has whole bits about what even is a cause. Like, we never see anything but A following B. So I think to a large extent what causality hinges on is an ability to intervene. So if you see children playing, and this is Spelke's work in developmental psychology, I think she's at Harvard now. They're often mini scientists. They're constantly playing around, pressing buttons, pushing things, and seeing what happens. So if you can do something reliably to induce a change in a system, then you can begin to infer causation from that. But purely from observation alone, it's very hard. So to make this a causality task, you'd have to actually allow participants to intervene in the sequential generation. But clearly they're very, very linked, right? And no one has yet. There's lots of ideas knocking around that sequential reactivation might be important for inferring causal maps, but there's no work on that yet. The more general question, though, is that sequencing, because we live in time, any the sequential relationship between experiences is often the way we build a non-sequential map. So the reason why hippocampus is important for episodic memory and for spatial navigation is because you need to experience. If I walk around a new city, everything I'm experiencing is just a sequence of one place after another place after another place. It is only offline when those sequences are replayed that you can furnish a non-sequential map. So they're not two separate functions; they are the same function. And there are now models of this, which I can point people to. But yeah. I don't know if that answers your question.
No, it does.
Free association.
No, it really does. And I guess on that note, is there a minimum level of replay that you have to see happening in order for the sort of phenomenal experience to have coherence?
I see. Well, we don't know if these replays are phenomenologically, whether they are. We don't know whether this is the neural substrate of mind wandering. Most of this work has happened in rodents during slow wave sleep, when it's not REM sleep, so it's not. There is dreaming which happens in slow wave, but it's not classically the slow wave, the dreaming bit of sleep. But certainly, if you disrupt replay in rodents, they don't learn very well about so that you put them in an environment, they walk around, you then get them to sleep, you can detect and disrupt the replay events, they can't navigate the environment as well. And you can control for generally just mucking around with the brain. But yeah, there's lots and lots we don't know about. It's a very controversial field.
Thank you. Just to go to a few questions in the audience. Got Lucy.
Thanks, Matt. That was a great talk. I was thinking about what you think about the transdiagnostic relevance of this, particularly sort of dementia where there might be cognitive impairment, how that makes this.
Yeah.
Could you just repeat that question?
So the question was about transdiagnostic relevance, and particularly relevance to dementia, where there are psychotic symptoms, but also, I guess, medial temporal lobe atrophy as well, and dysfunction. So yeah, it's unexplored. There's lots of work. So I think dementia and temporal lobe epilepsy in particular, these are two conditions which much more dramatically target the medial temporal lobe than psychosis does. So there's actually ongoing work. There are planned studies at UCL to investigate this kind of thing in early onset dementia. And yeah, transdiagnostic, if you think that this is a domain general mechanism for memory consolidation and for some kind, there's no relationship to symptoms here, so I don't want to stretch it too much. But if you were to speculate, then yeah, it would be a transdiagnostic mechanism. And certainly much of the word association stuff I've shown you has been looked at in dementia. There's a lot of studies, so if you have verbal fluency data in patients with dementia, we can model it very nicely these days. So lots of the same principles apply.
And a question from Pospo.
Yeah, it's actually one that's really straightforward, and the other one is a bit more of this. So the first one is about, so you showed that you've included both medicated and unmedicated patients. So number one, straightforward, did you see any difference? We didn't.
It's obviously halving the sample, right?
Yeah.
Also, because of the nature of my patients, the medicated ones were often the slightly more unwell ones. There's a reason why the unmedicated were unmedicated. So it's not a purely random sample, right? But no, we didn't see any difference. If anything, we saw stronger signatures in the medicated. sorry, in the unmedicated. But none of this is significant. Yeah.
And the second one, because I see Oliver up there in
Yeah,
the picture, and also wearing the IBPM.
Yeah.
So how does your findings align or take us further away from this whole theory about salience and the development?
Oh, I see. Yeah, that's interesting. So one of the salience theory of psychosis, for those who are not familiar, is it's very tied to dopamine. The idea that dopamine, the midbrain striatal or circuit, fires when there are unexpected things happening in the world, typically reward prediction errors, within the context of model-free reinforcement learning that allows you to learn about the value of states. So model-free reinforcement learning is, and the idea that Kshitij Kapoor said, what if this was going wrong in schizophrenia? You would then have your alarm, which tells you if there's something expected or not, faulty, which means you learn very abnormal sort of things about the world. There was something missing in that, and what's missing in that is the model, the internal representation. So reinforcement learning says that if I have box A and box B, I can learn about the value of box A by doing trial and error openings of box A and box B. And if I get 80% rewards on box A, I learn box A is better. I do that using dopaminergic reward prediction errors. But how do you get from that to complex representations of the world involving people, the relationships between them, causality? There's emerging work showing that dopaminergic prediction errors can help with that bit, but my view is that you need that. Salience is not enough. You can have a salience attribution model which says your model of the world is wrong, you need to update it. But how you update it requires something else, and I think that's where this is the distinction between, I mean, to use reinforcement learning terms, this is the model, the internal model of the world. So salience doesn't get you there. You need something else. So the current thinking, just to be very kind of simplistic, is that the salience attribution mechanism is still dopaminergic, but the map of the world that it's updating is in the hippocampus. So you have this relationship between the two, which I think Tony Grace has written about as well. But I think it's all related. It's all related. So dopaminergic reward prediction errors into hippocampus, change hippocampal, update hippocampal representations. So there's lots of work on that.
Thank you. We're just going to go on to one question from the chat. This is from Vasilisa Zhukova, who says, thank you for the talk. Could you please explain why you chose the pictures of the butterfly, the bananas, etc. for the experiment? Did you try to do the experiment with not abstract or somehow logical sequences of images? So that's a relationship between the
the thing we want from the pictures is for them to be as decodable as possible, so as visually distinct as possible, ideally, so that we can separate them. When those pictures are presented in sequences, which picture is first, second, or third is totally randomized between participants. So imagine you go into the scanner and you're like, well, obviously, butterflies follow marbles because of some weird thing you've come up with. Well, that's just going to be by fluke. The next person, they're not going to be related at all. So within a single participant, you can never be totally sure if they haven't got some preconceived notion of how these are related. But across the whole sample, when you randomize it, you can be fairly sure. So we want them to be as devoid of meaning as possible, or devoid of relational meaning. But there was no deep thinking beyond that, I'm afraid. And people have, that's what I would say on that, I guess. There's a whole bunch of experiments one might do based on what you just said. But,
Thank you. And then we'll just finish on
I think Paul had a question.
Okay, sorry, one question from Paul Shotbolt.
Yeah, thanks. It's a fairly poorly formed question, I guess, but maybe wonder about this idea that sort of thought evolved from movement.
Yeah.
I mean, it's lends a lot of weight to what
you're
saying, and in the sort of semantic maps related to maps of localization in space.
Yeah.
And by the same token, it really, for me, raised the possibility that sort of rodent or animal model for psychosis might be much more valuable than we thought because of this. Because essentially it's not much of a step to leave from problems navigating a maze to problems with semantic ability.
I'm so pleased that you come to that's perfect. There's a book called Metaphors We Live By, which you may have come across, written in the 1980s, which I highly recommend. It's kind of a cog-sci philosophy book, where the authors dissect out metaphors like, I want to get on top of a problem, or don't get ahead of me on this, and they just unpack the fact that what these metaphors, much of these seem to be grounded in, is in movement, bodily movement, or space. So evolution has bootstrapped abstract cognition on top of something that already existed, which was something which was absolutely necessary, which is getting around the physical world. And the same machinery has been leveraged. So yeah, perfect.
Yeah, so I also think there's a lot more in common between human beings.
Yeah.
And human
Oh, I see. Yeah, like obviously tick disorders and
exactly.
emitted movements and emitted thoughts and obsessions. Yeah. What is an obsession? It's fascinating stuff. Yeah.
And one last thing, I noticed you mentioned gamma oscillations.
High gamma.
High gamma.
Yeah.
That could potentially be a target for neuromodulation.
Yeah. Well, so again, good. I mean, so I think that if we think about neuromodulation, whether it's deep brain stimulation or the RTMS stuff that's going on at Stanford and Harvard, really impressive stuff. But I think to push forward, you need some model of how perturbing a neural circuit, even in a very localized way, causes some interesting cognitive phenomena. And I think that's my pitch for CogNeuro. One question which could always come from the audience is how is this going to help patients? Well, it's not tomorrow, but I do think understanding the relationship between brain and mind is kind of necessary for a mature neuropsychiatry. Yeah.
All right, thank you. I do apologise to Laith Alexander. We don't have time for your question. We are going to have to move out.
Laith, you can email me if you want.
But just really say thank you so much, Matt,
Thanks.
for a really fascinating talk. I think we're very sad that you're no longer
No, thanks for paying attention.
doing work
of
this quality and this fascinating.
Yeah.
We have our next meeting. I think it's in 2 weeks. Is that right? And kind of fortuitously sticking to the theme of the relationship between sort of movement and the mind, although in many ways changing theme, it's going to be on dance therapy for depersonalization disorder, if I understand right. So it's going to hopefully add some resonance to the phrase dance like nobody's watching.
Cool.
So we'll catch you. The adverts will be out for that soon, and we'll see you next time. Thank you. And thank you to Matt.
Thanks.